All right, let's dive right into this explainer. We are literally surrounded by living things all day, every day, yet the absolute fundamental building blocks that make up all that life, they're almost completely invisible to us. Well, today we are going to change that. We're going to bridge that gap between the world we normally see and the microscopic world we usually can't. But hang on a second. Before we even think about touching a microscope, we've got to ask a pretty fascinating question. Are these building blocks of life always invisible to us, or are some actually hiding in plain sight? You know, we hear the word cell and automatically assume it means microscopic, but nature loves to break its own rules. As we'll see today, size is totally relative depending on the biological job a specific cell has to do. Here's the game plan for today's explainer. We'll start with our goals and prep, take a close look at onion cells, switch gears to observe some fish egg cells, and wrap it all up with a side-by-side comparison. Let's get into it. Part 1. Goals and preparation. Getting our lab set up. You know, you really don't need some massive, multi-million-dollar facility to do real biology. With just a handful of simple tools, we can safely prep our specimens. We're talking flat glass slides, incredibly delicate little cover slips, some absorbent filter paper, a dropper, dissecting needles, a spoon, and a scalpel. Honestly, these basic instruments are all you need to unlock the cellular world. So here's our absolute essential checklist before we begin. We're going to need a microscope, one with a 40x objective is perfect, a simple magnifying glass, our slides, cover slips, and a little distilled water to keep our samples hydrated. Oh, and the stars of the show today? Just a totally regular everyday onion and some fish eggs. Moving right along to part 2, exploring onion cells. Time for some microscopic observation. What I absolutely love about this next step is how we take a completely ordinary kitchen veggie and turn it into a legitimate scientific specimen. If you just chuck a thick slice of onion under a lens, total darkness, light can't get through it at all. So first, we use our scalpel to score a tiny 7 to 8 millimeter square on the inside curve of an onion layer. Then we use a needle or tweezers to gently peel away the very top skin. That's the epidermis, and it is so ridiculously thin, it's practically transparent. Actually getting that epidermis off takes a little bit of finesse. You can see the technique right here. You score the tissue and then use a steady, gentle pulling motion with your dissecting tools to separate that single cell-thick layer without ripping it. It definitely takes a steady hand. Okay, so once we've got that ultra-thin layer, we have to mount it. We drop it onto a slide with a little distilled water just to keep those delicate tissues plumped up and happy. Then comes the tricky part. You hold the cover slip at an angle and slide it down super carefully. Why the slant? Well, if you just drop it flat, you're going to trap air bubbles. And trust me, under a microscope, air bubbles look like giant, distracting black donuts that totally ruin the view. Finally, just dab the edges with some filter paper to soak up any extra water. Moment of truth. We pop that slide onto the microscope stage, get it in focus, and bump it up to the 40x objective. And wow, just look at that. This breathtaking, highly organized structure pops right out. Doesn't it look exactly like a perfectly laid brick wall? There are literally thousands of individual units, all packed tightly together to give the plant its structural integrity. Looking at this biological brick wall, we really want to identify 3 distinct parts. First, see those thick, dark borders? Those as the cell walls. They're what provide that rigid structure. Second, that dark little circular spot sitting inside, that's the nucleus, basically the cell's genetic command center. And third, the fluid-filled space taking up the rest of the room is the cytoplasm. All right, on to part 3, observing fish eggs. Let's look at some macroscopic animal cells. We are making a massive shift in perspective here, zooming way out from the microscopic back to the macroscopic scale. Prepping these animal cells is so much faster and gentler than the onion. You literally just scoop some fish eggs into a petri dish with a spoon, add a splash of water so they don't dry out, and use your dissecting needle to very, very gently tease them apart. If you've ever seen fish eggs, you know they naturally want to stick together in this protective, jelly-like clump. But look how vibrant, translucent, and perfectly spherical they are once we start separating them out. And here's the absolute kicker. We are totally bypassing the 40x microscope for this. Remember our question earlier about seeing cells with the naked eye? Well, here is your answer. Every single one of those unfertilized fish eggs is technically one single biological cell. They are so massive that you can see their basic shape, color, and boundaries with completely unaided vision. Or, maybe a basic magnifying glass if you want to catch just a little more detail. Which brings us to part 4, comparison, and conclusion. Let's analyze our data. Let's bring all this together. If we map out exactly what we could and couldn't see, it paints a pretty clear picture. With the microscopic onion, we could see the whole setup: cell wall, nucleus, and cytoplasm. But with the fish egg, since we're just using our eyes, we can easily see the outer spherical boundary and the cytoplasm. But that tiny nucleus stays hidden without some extra magnification. So the big takeaway from today's explainer is really this wild visual contrast between the two kingdoms of life. On one side, the plant cells, the onion. They're rigid, rectangular, totally dependent on a microscope to be seen, and locked together like a brick wall. On the flip side, the animal cells, the fish eggs. Soft, perfectly spherical, huge enough to see by just looking at them, and floating around as squishy, separate, individual little units. I'm going to leave you with a quick thought experiment to test out your newly minted micro-explorer instincts. If I were to hand you a completely mystery slide right now, and you peered through that lens, what is the single, absolute biggest visual clue you'd look for to figure out if you were looking at a plant or an animal? Think back to the brick wall versus the soft spheres. Keep exploring, keep asking those big questions, and I'll catch you in the next explainer.